Hello students, in today's class we are going to learn about chloroplast isolation by sucrose density gradient centrifugation method. So the aim of our experiment is to isolate cell organelle, that is chloroplast, from plant tissue using the chloroplast isolation buffer by sucrose density gradient centrifugation method. Requirements for the experiment includes fresh leaves of spinach, chloroplast isolation buffer, mortar and pestle, muslin cloth, and other lab equipment. So chloroplast is basically the organelle that is present in plant and algal cells that carry out the process of photosynthesis. It utilizes sunlight in combination of water and carbon dioxide to produce sugars for the plant. Chloroplast is surrounded by two membranes. The outer membrane is permeable to small organic molecules, while the inner membrane is less permeable and is studded with transport proteins. The innermost matrix of chloroplast is known as stroma, that contains the metabolic enzymes. It also has a third internal membrane system, which is known as thylakoid membranes, that contain the light harvesting complex, whereas the photosynthetic pigment, that is chlorophyll, functions to capture the energy from the sunlight and converts it into ATP and NADPH that are utilized to make organic molecules. They carry out number of other functions as well, including fatty acid synthesis, amino acid synthesis, and immune response in plants. So all green parts of the plant contain chloroplast. Chlorophyll in them make the photosynthetic part of the green plants, and there are approximately about 50 chloroplasts that are distributed per plant cell. Coming to the principle of the isolation method, so first of all, it involves breaking of the cell wall mechanically by making use of a blender. The unbroken leaves are then removed by filtration process through a double layered muslin cloth. The filtrate that is so obtained is centrifuged at about 1000 rpm at 4 degrees Celsius to further remove the debris of small broken leaves. The supernatant then obtained can be further centrifuged and then it is finally passed through the sucrose gradient and followed by further centrifugation. A distinct green color band that is obtained in the gradient is recovered for the further assay. So during the experiment of isolation of the chloroplast, the disruption is facilitated of the cell wall by mechanical shearing. Sugars or alcohol are used in the medium in order to maintain the osmotic concentration inside the cell. Sorbitol is added during the isolation to prevent the cell from rupturing. DTT is added as a reducing agent and it helps to prevent enzyme degradation by oxidation. MgCl2 ion increases the molarity of the buffer solutions containing cells, making the cell environment hypotonic, thereby making cell lysis faster. And the monobasic and the dibasic salts used in the experiment are because of their higher buffering capacity. Coming to the procedure, so first we need to prepare the chloroplast isolation buffer working solution that has a concentration of sorbitol about 0.32 molar, MgCl2 5 millimolar, DTT 1 millimolar, sodium phosphate monobasic 5 millimolar, and sodium phosphate dibasic 5 millimolar concentration. Then we need to weigh the 20 grams of spinach leaves and wash them 2 to 3 times with distilled water. The leaves are then dried on a blotting paper and are ground in 20 ml of freshly prepared CIB buffer using mortar and pestle. The homogenate so obtained is then filtered through double layer of muslin cloth and the volume is adjusted to 50 ml with the CIB buffer. The filtrate is then transferred to a centrifuge tube and centrifuge for 5 minutes at 4 degrees Celsius for 1100 rpm. Then to obtain a clear green supernatant, a further 5-minute extra centrifugation may be required. The final supernatant obtained is free of cell debris and it is stored at 4 degrees Celsius. We then need to prepare 2.5 molar, 1.5 molar, and 1 molar sucrose solution in CIB buffer. We then need to fix the falcon tubes on the stand and add 3 ml of 2.5 molar sucrose solution into the bottom of the tube. Then gently the second layer, which is of 1.5 molar sucrose, is added on the previously poured 2.5 molar sucrose layer. Again, along the walls of the tube, finally 3 ml of 1 molar sucrose solution layer is added on top of 1.5 molar sucrose solution layer. Finally, at the end, 3 ml of spinach leaves homogenate is added on top of the gradient. This is followed by centrifugation at 3300 rpms for 30 minutes at 4 degrees Celsius. The falcon is then removed from the centrifuge and observed the obtained layers are further analyzed. Coming to the observation, after density gradient centrifugation, as you can see, 3 different layers are obtained. The top zone is slightly brown in color and it is indicated of presence of free chlorophyll. The middle dark zone or the dark green zone indicates presence of chloroplast and mitochondria, while the bottom bright green zone indicates presence of lyse cells and lyse chloroplast. As you can observe, the chloroplast and mitochondria fractions are obtained between the 1 molar and 0.5 molar concentration. So 1 molar is the is present till the 9 ml height, and above that is the 0 molar. So in between we find the layer of green colored chloroplast being isolated. So coming to the precautions, we need to carefully prepare the sucrose gradient, and one should be able to clearly see the interface between the layers when we overlay one sucrose layer above the other. All the glasswares and the apparatus should be clean and dry. The midvein of the spinach should be removed before grinding, and DT2 should always be added freshly to the chloroplast isolation method. So that's it about chloroplast isolation method by sucrose density gradient centrifugation. Thank you very much, and kindly like, share, and subscribe if you like the content.